Welcome back, guys. Today the topic was lipids, or better known as fats. So we're going to take a look at a bunch of the different parts, the job to perform, the physical characteristics, dietary recommendations, and then the process of dehydration, synthesis, and hydrolysis when you're talking about lipids. So a few more jobs on this one. Obviously, the biggest job that fats are known as are sort of a long-term storage of energy. And the biggest way that they store are these things called triglycerides, which we'll deal with here in a little bit. One good little fact here to know, which is it's kind of interesting, is that 1 gram of lipid equates to 9 calories. And if you remember from the video yesterday, carbs, 1 gram is 4 calories. So even though carbs are your preferred method, fats themselves actually contain more calories. You can think of them being just a little harder to get to, I guess would be the best way to say it. So storage of energy is a big one. They do protect the organs. Obviously, when you have lipids or when you have fat over the organs, there is some measure of protection there. So some measure of insulation. Obviously, living in Minnesota, it gets cold. If you have a little more lipid or fat, it's going to be insulation, it's going to be some warmth. They do create biological membranes. It's a term you're going to hear again multiple times, but it's called a lipid bilayer. It's made of a special type of lipid called a phospholipid, and that is what actually makes up all of your cell membranes, the boundaries which keep your cells. They keep them intact. It may do or will do, there are some chemical messengers, some steroids is one aspect of lipids as well. And lastly, they do work to absorb some of the fat-soluble vitamins which you intake in your diet. Without fat to have them dissolve in, you obviously would not be able to get those, so they absorb those as well. So that's just to look at some of the jobs that are being done by fats. The next topic is to look at different types. Now again, we already mentioned steroids, but we also have to just briefly talk about cholesterol as well. And people have heard cholesterol in terms of bad cholesterol causing some different heart diseases and things like that. So we'll take a look at that a little bit. We also have what are called phospholipids, and there's a structure of a phospholipid there. This, again, is what makes the actual boundaries of the cells. It's actually made of 2 different parts. These parts down here are called fatty acid tails. I'll just put them as FA, fatty acid. And then this top part up here is a phosphate head. So it's a unique little thing, typically drawn in the book. It almost looks like a jellyfish. There's the head and then the 2 tails. And actually in your body, when these things line up, they line up in what I called a bilayer before. So they literally will have head and tail, head and tails, head and tails of one side, and then the other side will be just the opposite, like this. And then that will actually create a barrier for your cell membrane. It creates a waterproof, essentially, barrier. Okay, and then the third, and this is the one we're going to focus most on, are these things called triglycerides. If you saw me in class talking to people, I refer to triglycerides. I say they look like a big E, where the backbone, this part, which is also this part, is made up of something called glycerol, and then the 3 other parts of the E, if you will, are the fatty acids, and they can be in different forms, either saturated or unsaturated, so we'll get into those. But so you have a glycerol backbone and then 3 fatty acid chains, which are all of these things here. So 1, 2, 3. Or chemically, that's what they look like. Oh, that kind of gives you an idea of just general types. Now taking a look at the physical characteristics, and this is where it gets a little more detailed. As far as that E goes, it's made up of C's, H's, and O's, which are just like carbs, right? Carbs, if you remember, from yesterday. When you're talking carbs, it's C1 to H2 to O1. So it's that 1 to 2 to 1 ratio. Right? Now, for lipids, so if we do lipids on this side, it's C's, H's, and O's. And I like to do it this way. I say it's a lot of those to a lot of those to very little of those. So you might get these in a 1 to 2 to very little ratio. So let's say it was like C10. It'd be like H around 20, O like 2. That would be a definite lipid. So a lot to a lot to a little. Okay? Beyond that, let's take a look at the actual characteristics. You are going to have two different types when you talk about characteristics. You're going to have what are called saturated or unsaturated fats. Saturated fats are the ones that are liquid at room temperature. These ones can be in different forms. So you can have cis fats, you can have trans fats, but these are unsaturated. So they are, and we'll do some diagrams here on the next slide to get a picture of what they look like. But these are the fats. You can have cis unsaturated fats, which are good natural fats, and then you have trans unsaturated, which are a little less that you want. Those are the ones that lower your good cholesterol, and they kind of raise your raise the bad type of cholesterol. And we'll talk a bit more of that here too. Version obviously is saturated fats, which are solid at room temperature. If you had to think as a general, these are bad, these are good. Okay, these are the ones that come from animal sources. Things like cream and cheese, and you get into some of the real fatty meats. That's what you're talking about when you're talking about saturated. Unsaturated would be things like oils, vegetable oils, things like that. When you look at these guys, so when you look at these, you're going to end up getting some sort of different ideas as to how the thing actually looks. So what I'm going to do here is just open up a clear slide and show you kind of how the unsaturated versus saturated things look. And we'll draw some of the structures for them. Again, remember, it looks like a big E, and that's the key. That backbone of the E is what's called glycerol. So it will actually be a structure that has a carbon, and a carbon, 3-carbon chain, and then off of those carbons there are H's to fill in the majority. And remember, with the honk rule, carbon needs 4 bonds, and off of each of these there will be a hydroxyl group. So that's the backbone. That is glycerol. Now, when you make the actual fatty acids, so I'll do the fatty acids over here. Let's start with unsaturated. Fatty acids are going to be a chain of carbons, all right? And depending on what type, it could be a long carbon chain. I'll draw one that maybe has 4 or 5 here just to show you. But so we could go 1, 2, 3, 4. Let's go 5. So I have that. Now, to be a fatty acid, the acid part of it comes back from the functional group on this end. It's called a carboxyl group. All right? So it'll have a double bond to an O and then a single bond to an OH. That is the carboxyl group. Then on these carbons, if we make this guy unsaturated, that means that there has to be some double bonds between the carbons. Okay? And I'll show you why here in a second. So let's put 1 here, and we'll put 1 here. Okay? Now, according to the honk rule, each carbon needs to have 4 bonds. So this one is lacking 1. So let's add a hydrogen here. 1, 2, 3. This guy is lacking 1. So let's add a hydrogen here. 1, 2, 3. This guy. And then 1, 2, and then this guy. Notice there are, down through here, there are no hydrogens. And that means this is unsaturated. If it was saturated, there would be a hydrogen everywhere you look. and there would be no no double bonds. Okay. Now that is actually a type called unsaturated, but it has a even more specific name. It is called and if we actually named everything, this would be a cis unsaturated or poly unsaturated fatty acid. The cis comes from on either side of the double bonds. So on this side and this side, the H's are on the same side of the double bond. So cis comes from that. They're like that the whole way. It's unsaturated because there aren't H's everywhere, and it's poly because there are more than one double bond. If there was one double bond, it would be a mono-unsaturated. So again, a lot of the nomenclature comes in here, a lot of the naming stuff. The other, so that's one, and then we could do another one, because there's going to be three of them. There's going to be one that's we're getting ready to attach here, one to attach here, and one to attach here. So that one is a cis polyunsaturated. So we'll leave that there. The next one I'm going to do is going to be, so we'll go the same. We'll go C, double bonded to an O, to an O, to an H. And then we'll have 1, 2, 3, 4. And I'll even put the double bonds in the same spots. Alright? Now, this is also going to be unsaturated again. But this one we're going to make what we call trans polyunsaturated. Now, trans, if cis, they exist on the same side of the double bond, then here they're going to be on opposite. So I would put my first H there just like above, but this one would go below. Okay, let's say this one is below again, and this one would be above. So it is trans because the H's are on opposite sides of the double bonds instead of being on the same side. So it's a trans because of that, poly because there is more than one, and unsaturated because there is not an H everywhere you look. The terminology gets really heavy on this stuff, but if you know the words, it can make sense to you. And then the last version then, so if those are both unsaturated, then let's look at a saturated fatty acid. Okay, again, we have our C, our O, our H, and we go 1, 2, 3, 4, I think 5 is what yeah, 5 there. Now in this one it's easy. There are no double bonds, so to make all of the carbons stable, I would add a hydrogen at each one, just like that. Alright, so that would give you the different types, the polyunsaturated, the cis polyunsaturated, the trans polyunsaturated, and in this case the saturated. Alright, that's a lot of stuff going on, but that shows you how the different fats work. In terms of what is better, what is worse, remember that unsaturated tend to be the better of the, if you have to pick between saturated and unsaturated, unsaturated is better. And within the saturated, within the unsaturated fats, trans fats are the bad ones. Most processed foods are made with trans fats. It makes them stay on the shelf longer. They don't rot or they don't go bad as fast. There's a bunch of reasons why. It has to do with refrigeration and all kinds of stuff. All right, so that kind of spells it apart. Now, you get the idea there, but now let's actually look at the thing and see how the thing actually. So the last part is to just look at the process of hydrolysis and or dehydration synthesis. So we'll look at it in terms of a triglyceride. So what you're seeing here, they're both here actually. It's the same drawing for both. Remember, if you look right down here, dehydration synthesis is going this way. Hydrolysis then would be going up in this diagram. And essentially, it's the same as it was with carbohydrates. All you have to figure out is where it actually occurs. And in this case, if you look, to do hydrolysis, you have your big triglyceride, your big E right here. Okay? And what happens is that between the purple and the blue, if you look at the diagram, so right here, and right here, that bond is going to be broken when you do hydrolysis. Remember, hydro means water, lyse means to break. So you literally add this water, and once you've added that water, those bonds are going to break right here, here, and here. They're going to split. The H will go along with the O. The OH will connect to the carbon. You will get your 3 fatty acids, and you will get your glycerol separated by hydrolysis. When would hydrolysis happen? That would happen during digestion when you ingest a fatty acid, a triglyceride, glycerol with fatty acids. You need to break it down to use the energy that's stored up in there. So you actually would go through hydrolysis. The other version, the other version is dehydration synthesis. You dehydrate, so you minus water. Synthesize is to create or to make. So you basically reverse, and now you have, and again, this would happen if you ingest some fatty foods, you break the triglycerides and the fatty foods down into the individual components. You don't use them, you sit on your couch, you're eating potato chips, you're just hanging out, not doing much. You don't use the energy over time, and it obviously takes a little while for this to happen. Days, right? First you'll use up your glucose and then your glycogen, and then if you don't have enough intake of those, you then go into the breaking down of the fats. But to make the fats, to put that extra energy into storage, you take your glycerol, you take your fatty acid, and you remove water. And you can see it right here, it's in circle. H and the OH, H and the OH, each one of those. You remove those, that creates a bond between the O and that carbon, which you see right there. And once you've got that bond in place, you have a completed triglyceride with 3 different water molecules, 1, 2, 3, to make that thing. Okay, so if you look at it, that should be pretty, again, because you know it from the carbs, this shouldn't be all that much different. But you now can tie that in, along with what you know right now on the different types of fatty acids. You can look at the saturated versus unsaturated, like right here. Hopefully you guys are looking at these diagrams here as we're talking. This guy is saturated because there's no double bonds in the C between the C's. Same here. This one is a mono unsaturated, and you could actually tell me what kind, because the H's are on the same side of the bond. It would be a cis. So, just again, every time you look at it, think through those ideas. Use these terms. And then the last thing I put on here, this will be the last slide, is just some basic dietary recommendations. Typical American diet, or typical diet, they say you should max out at about 30% fat. Low fat or no fat diets are not very good diets. You need a certain amount of fat. Of that 30% max, only 10% should be from saturated. The rest should be from unsaturated, and preferably cis unsaturated. And then we also mentioned earlier that 1 gram of lipid equates to 9 calories, whereas carbs, 1 gram of carbs equals 4. So that's just a quick review. Hope you found it useful.